I will go back a step now and actually show you a little bit of what are iPS cells and how we can use them. So yeah, iPS cells, they're a hype in the moment. They're in the literature, in the press, everywhere. You will have seen probably the pictures. So this is Professor Yamanaka and also Sir Vernon, which was also named for the kind of very early work on reprogramming using amphibian oocytes. And mostly together with these naming comes this beautiful picture of the spring of use, which is hanging in Burning actually. And basically there's a claim that now that we have iPS cells, we have like also reversed aging because we have reversed the state of the cell. And I'm not so sure that that is true. It's probably more a selection like we have in normal fertility, yeah? So not every sperm and every egg can produce a healthy living baby. So there's a really tough selection there for the youngest cell ever. And I think that's the same happening in iPS cultures. So can we actually use them? Will they be of any use to us in the aging research field? And I would say yes. There is some knowledge here to be gained of using these cells because you can have them from different disease backgrounds, and that was not available when we had the human embryonic stem cells. But now we can really get them from every disease type and every genetic background. So the other step is you can actually make interesting cells out of these iPS cells. And that's also something which was missing in the aging models. It's, here we have fibroblasts and we have used them as in vitro models. We have also endothelial cells, but we didn't have like all cell types of the body in the dish. Also we can study kind of rejuvenation effects, which might be part of the reprogramming. And yes, we can probably use them to study aging or aging-related disorders in addition. So to take that together, these are the classical models used in aging research. And what we have now here is additional model is using the iPS cells and then do differentiation in all kind of cell types we want to study. And we can even do transplantation studies, or we can form them into organs or tissue combinations, which has also not been done much in the aging research, like studying how, for example, cardiomyocytes and fibroblasts age together. And we can even transplant it back into a mouse and kind of have a pseudo in vivo environment. But we have to be careful. So to know how your iPS cells are in a model, you have to also look at, we use all kinds of donor tissue in a moment. So the age of donor might have an influence on what you will later see in the dish. We have to look at genetic background. So in the moment, we don't really look at the genetic backgrounds of our iPS cells. And yes, that's obvious. If you have a diseased iPS cell, that will probably behave differently. Also, there is no common factor set in the reprogramming area at the moment. So there are lines around which have used 6 factors, 4 factors, the 4 Yamanaka factors, or the 4 Thomson factors, and now additional factors are coming out. So also that might have an effect on how your iPS cells as a model will work. And also, remember, these are mostly virus reprogrammed iPS cells. So I will have integration of the virus if that was a lentivirus or retrovirus. So we don't know where they will integrate and how that will affect, again, how the iPS cells behave in a tissue model. Also, dosing is really very, very different. Sometimes we have 5 integrations of a virus. Sometimes we don't have a shutdown of the virus. So we still have a permanent OCT4 activity in our lines. So that's also something we have to really control. And there's also lines which I would call partially reprogrammed, which show some signs of pluripotency, but for example, they have not formed teratomas in vivo. So how do we kind of control for all of these factors? And then yes, the beauty is you can really study aging during the reprogramming phase, and then also kind of like in the early development during differentiation. So you can look at kind of like what is not in a moment really possible in humans is like early events. You can have effects of like what is mutation doing early during differentiation, later on when you have formed the cells, and then they start to miss. So this is kind of like the beauty in it, but again, you have to be very careful of the cells you're using. Nevertheless, there are now labs everywhere having established kind of the differentiation protocols that were all cells derived in our lab from virus iPS cells. And today I will just briefly show you a little bit of data about the fibroblasts. So we actually have fibroblasts as like starting material, made iPS cells, and then also differentiated them back into fibroblasts. So there was some kind of literature showing that the iPS cells, very similar to the Dolly effect, show premature aging signs, so kind of much older than their donors. And we wanted to have a quick look at how true that is. So this is our starting material, primary fibroblasts from the dermis. These are then our iPS cells. And again, we show that when we differentiate, we have really mostly fibroblasts. We then do the usual stuff. We go through culturing these cells and looking at the passage levels. We control for beta-gal staining, and here's just one showing that there is, with time, really senescence. We don't know yet if that will be different or not, because it's kind of like not enough lines. And this is showing you one of the problems we actually have here. So these are 4 different donors, so 4 different iPS lines, and you can already see we just measured P53 here. So already the starting material is quite different from each other. So that was just like the mean of all donors. So we don't really know, like if we compare that now with normal aging, if we start with something like this, we might have completely different behavior in the cell culture than if we start with iPS material, which has very low P53. But fortunately, so this is here the donor fibroblast, one example, just showing like them on different passages at P21 and P53. So it's upregulation here. And you mostly also see that the iPS fibroblasts also show this upregulation. We heard that during reprogramming, telomerase activity is actually activated. So again, if you look at that donor fibroblast, iPS versus iPS fibroblast, happily the iPS fibroblasts also downregulate telomerase activity. But we don't know what effect will be like if you expand your iPS to a very high level, because we see here an increase in telomerase activity, and we have not yet started to differentiate these ones. So again, you have to be careful, like, would you study material derived from an early iPS or late passage iPS, and is there a drift here? So kind of like, be careful. So we started with very early iPS cell and differentiated them. So it was actually passage 2 which we used, because we have really otherwise great problems to derive fibroblasts. And then if you here look at later fibroblasts from these iPS cells, you also have like a downregulation or like a shortening of telomeres. And we have to really follow this now if we really get like to the same end results. And also one problem might be here that we have contaminated iPS cells left in the culture. I mean, we try to be so good in the differentiation that we really have like 100% fibroblasts and nothing left. But the selection is not yet so perfect that you can be absolutely sure that you're not having any contaminating iPS in the culture. Again, some groups have described that mitochondria also go through a phase of reprogramming / rejuvenation, and that there's actually a reset of mitochondria. But I don't know how true that is. Is that, again, mostly the effect of selection? But they do have shown that there are differences. So basically also looked at mitochondrial morphology to start with. So this is just like the donor fibroblasts. These are the iPS cultures then, and that's fibroblasts derived from the iPS cells. And basically, they're very similar. If you look at just that size, you see, yes, in the iPS cells, they become smaller. So there might be kind of some regeneration going on, but maybe it's just selection. And also, if you look at membrane potential, here we included some other donor lines. You see again that the iPS cells have a slightly higher membrane potential, but that's decreased again in the iPS fibroblasts, similar to the original donor fibroblasts, which is nice. We also looked at data coming really from the literature, and these are 11 publications on reports on either telomere lengths or mitochondrial activity, all looking at if there's pre-senescent phenotypes in the cells derived from iPS cells. And basically, now that we have analyzed all that data, we can fairly sure say, no, there is in proper reprogrammed iPS cells, there is no such thing as a pre-senescent phenotype. So they behave very much like their donor fibroblasts cells. And Claire and Carsten did most of the transplantation work. Then thank you very much for your attention, and hopefully there are questions.
Thank you very much for your interesting lecture. Alexandra, are you familiar with the works of Marek Ratachniak? If I named his second name correctly. Are you familiar with his works about such subpopulation of pluripotent stem cells in adult organisms, very small embryonic-like stem cells?
Oh, yeah, basically the concept, yes, I know the concept.
Yes, so maybe it is more perspective to know how to use that, because as I remember, I do not know. Of course, it's a pity, but I don't know fresh works and fresh publications. But elder one said that even aged organism has a large amount of these cells. Maybe it is perspective to use to find possibility to use them.
But yes, the publication is showing that these pluripotent stem cells still exist in the aged animals. I think it was only animal work which was published on age-related numbers. But they have not shown any function of these cells, so numbers is not telling me everything. Unless somebody is transplanting them in a disease model and shows the old ones are as functional as the young ones.
They function, of course, they function the cleans, but as I remember in his publications, he told that the amount in bloodstream after ischemia of heart, if I'm not mistaken, the number increases in bloodstream.
If they're of good quality, there's nothing against using these cells, yeah.
Okay, thank you.
Other questions? No other questions?
So, where do you think this kind of work will become widespread? Because, as I understand, it's a laboratory here and a laboratory there. When do you think it will become, common access?
Well, Yamanaka wrote that he would be starting a clinical trial using iPS cells next year. So I don't think that it's just one little lab here and there. It's really.
No, I mean, when does it get to the pharmacy? To the pharmacy, to the apotheke. When do you see this in the apotheke? In the clinic.
In the clinic? Oh, you mean in a clinic? Well, as I said, like Yamanaka wants to start next year, and then it's phase 1, and then you have to wait until phase 2 and 3 is finished, and then probably you can buy it on the market. So it will take definitely 5 to 10 years.
So with those IPS, there are many applications that we see. That's why everyone is just amazed by this. Concerning aging, there is a startup that most people on the internet know in Canada that tries to extract the secretions of those cells to sort of rejuvenate some tissues. Is that one application that you think may, because you've studied a lot what the stem cells become when we try to differentiate if they look like normal cells or not. So do you think that there is some rejuvenation secretory factors that may be compatible with the body?
Well, at least in the cell culture dish, people have looked at conditioned media derived from embryonic stem cells or iPS cells, and they have shown some reprogramming, rejuvenation effects. But that's only looking at it in a culture dish. There's some trials looking at MSC-secreted phenotypes or kind of like substances, and that has been applied in humans, but just on skin, on aged skin. So yes, it's a possibility to use it this way.
Thank you very much. That was very interesting. Do you perhaps know, or have an idea or have a hunch on why your microglia, the iPS-derived microglia, are not that efficient? Maybe it's because of the reprogramming method. Are you, will you test any other, like Yamanaka cocktail, any other methods of reprogramming? Or
well, I think we have to just get the microglia into the right state, and there's not much known of like kind of what factors to apply to the microglia to have them in the right status, because microglia can be everything from rested, semi-activated, fully activated, pro-inflammatory, regulatory. So I think we have to spend a little bit more time on the differentiation protocol to get them into the stage which might be useful.
Okay, no further questions?